monitor, and I might email it to Erica Burton to say, do you know what this is? Because I didn't see it in either the Ambari guide or the Sir Ridge guide. Or the Davidson guide or the Sir Ridge guide.
Yeah. Thanks for giving us the spelling of the Calyptrophora and Narella. Thank you.
Oh, you're
I was
welcome.
pretty close in my, yeah, Calyptrophora.
I always forget that R. I always have to remind myself there's an extra R in there.
The part?
Yeah.
Yeah. Ooh. Here we have another, what is it on? A little piece of rock there.
Yeah.
Get a bit closer, a bit bluish.
So the bentacodon jelly, is that jelly that the red one that we passed, Taylor Ann?
Yeah, the ones that swam across the screen for a bit, yeah, bentacodon.
Yeah, okay. And then I'm looking at your data entries here. What was I looking at? The Tromicosoma, those are the
oh yeah, those are the urchins. So there's two,
okay.
the yellow ones and the purple ones. So I'm not sure what the yellow one's like name is. I know they're both Trochosoma, but I think the purple one is like Trochosoma pana something, Panamese.
Mm-hmm.
But I'm not sure. Panaments. But I'm not sure about the species name of the yellow ones or if they're the same.
And these long tentacled anemones, I guess they are?
Yep. Deep sea anemone of some sort.
Mm-hmm. And the fish here is is it just a grenadier?
Yeah, it's just a grenadier, just a view from the top.
Yeah. I guess with the white head, it just, I sometimes think it might be something else, but yeah.
I think these anemones are Bolosera that we're seeing.
Oh, the one with the long tentacles, Bolosera?
Yeah. And early in the dive, when we were out in the mud and saw that white puffball pom-pom anemone, that is the one that's the tumbleweed that will actually tumble and move like we were talking about.
Mm. They're often white then?
I think so. I just happened to go by it in the deep sea guide I'm looking at here, and I was like, oh, look, it is a tumbleweed anemone.
Yeah, I've only ever seen it once or twice before, the ones that actually roll around, and there was quite a current that was taking it, if I remember right.
I guess I should talk to Erica Burton about how to put together a ID guide for the Santa Lucia Bank. We should start working on that.
Yeah, and we'll be.
I'm not saying.
Oh, go ahead.
No, go ahead.
As I say, we're working on an ID guide for deep sea corals and sponges for the West Coast initiative that the Deep Sea Coral Program is doing right now, and we'll definitely be incorporating some of the images from around here in this guide.
Pretty cool. Oh, you were telling us about that, and you're working with the Alaska Fishery Science Center because the next initiative is going up there. And so we'll have a guide that spans the entire West Coast.
Yep, and we also have some colleagues at DFO Canada who are contributing as well. So it's going to be the whole Northeast Pacific coast from the California border all the way up to the Aleutian Islands.
We worked with DFO a few years back. Good group. It's a Bowie seamount.
Yep.
We remapped that one with backscatter. It's really cool.
Yeah,
I think that Liz has been there as well, right? To Bowie, to Cobb and Bowie. Maybe I'm wrong. But...
they might have gone to Bowie on that first expedition. But I know in 2018, Nautilus did the Seamounts expedition with DFO Canada to Bowie, Skon-Ginglas, and a couple of the other seamounts up there. I'm actually working up that data right now.
Nice. Yeah, I was doing
the genetic
mapping
data
on that one.
Yep, the genetic data for the samples that were taken, and then the eDNA data to pair with the DFO surveys.
So with the eDNA, can you explain what types of DNA? Is it mitochondrial or what sections? Sort of what is the technique or that is used?
Sure. So first, just for background, for anyone who's listening along at home and watching along with us, eDNA stands for environmental DNA. And what that is instead of taking a tissue sample from every single animal in the area, I just capture a water sample, and that contains, presumably, all of the cellular material that all these animals are shedding into the water column. And we can extract DNA directly from that. And it gives us a picture of the local biodiversity. And what I'm using are actually mitochondrial markers. Right now, I'm looking primarily at the fish community as well as the octocoral community. And so I have a set of DNA primers that will amplify octocorals and are specific to octocorals and for fish. And so I then run those through a high power sequencer, and I compare them to reference libraries that I've built, and I'm able to identify all the animals that are either corals or fish in the local community. We can expand that and look for other types of animals. We can look for invertebrate crustaceans, but since I'm working with the coral program, we're interested in corals and then the fish that are associating with those habitats.
Thank you. And is the sequencing, is that now fully automated?
That's an end of transect right there.
Okay, thank you. I was wondering. I'll make note of that.
It's the sequencing itself, once I get it on the machine, is automated. I still have to do all of the DNA extraction and library prep for that. So I make the libraries that we run on the sequencer and do all the extractions in the lab.
If you can just pop in and take a look at HiPAC for a second, as long as, is that what you have up on channel 3?
Yep, it's on channel 3.
Okay, great. Yeah, so right now you can see our ship course We've been just kind of going straight to 5G. As long as that's still okay, we'll just keep on going in that direction. I don't know if you wanted to pop down to 5F or follow a long contour or just kind of go straight to any of them. Kind of seem, 6 to 1, half a dozen either. But I don't know. Let's see the timing of the dive where it's quite a ways to 5G. And
Yeah.
Sorry, I just tugged it.
Okay. If we can at least make it through that bright orange area.
Yeah, that was my thought when I had said maybe go straight to 5G, was just trying to get into that,
Yeah.
really high relief area
Right.
right there.
yeah.
Yeah, maybe hit the bottom part of it and then we can go up through it or zigzag through it.
Sure, well,
Yeah,
we
that
can
would-
just kind of head, I don't know if we wanted to, we could come down here, and then just kind of go along that, like this, if you wanted.
That,
or we
that
can
sounds
just,
great.
yeah, that way we'll get maybe a bit more time with it.
Mhm.
So I'll
Yes, that's,
I'll
we want to
redirect
maximize our
course
time.
then. So I'm going to change course to 135.
135?
135. Yeah, you're quite laid back, like 150 meters or so from the
Okay.
ship. So it might take you a bit of time to feel that. But bridge nav. Change course to 135. Thank you. Yeah, so that should be a little more along slope
Okay.
rather than up it. We're kind of like cross-cutting it, but we'll be going along.
Okay. So kind of the same thing, but
Yeah, mostly the same. Not too much of a change, but we'll eventually be going. See how like in Argus they're going the way that slope is, right? Instead, we'll just be going along it rather
Okay.
than into it.
Well, we found some rocks for our transect. Now we're back in the mud.
Yeah.
Yeah.
What's interesting though is I took just a peeked again at the backscatter, and in that area that we just were, it's really not. There's nothing indicative that that would be any different from what we're seeing now. I think just the resolution of backscatter at this depth,
Yeah.
with this how sparse those rocks were, it's really just not significant.
Yeah.
Yeah, and then it's just also it's hard to tell. Like the first dive that we did on the Santa Lucia bank, the Palm dive, that was just bright white. And yes, it was all hard rock, but it was that cobbly rock.
Right. So a lot of it is also relative. So we're able to kind of scale that when we make it.
Can you move that box a little so I can see the lat long, please?
Roger. There you go.
Well, I just have so much respect for mapping and the interpretation and just the processing, the stitching together of all of the different swaths that have been mapped
Yeah, that's
of this area.
and that's the secondary stage of processing too. The first stage is I spent a lot of time removing a lot of individual XYZ points from a cloud of points. Try to get it to
It just seems like an art.
a bit of that. Yeah, it's also it's super helpful to view it all in 3D when you're dive planning and stuff like that. So it's kind of a shame that it's nice to have the scientists out here to do that. So I think we're gonna work through some workflows to be able to do that.
Ooh, there's another one of the okay, Tilleran, hold on. Benzocodone.
Yep.
Yeah. So after this, I have to come out again and do something because all this knowledge,
Yeah.
got to use it again. Use it or lose it. And Taylor, and how, this is your second year, how did you come out the first time?
The first time I was an ocean science intern last year, and I came out for 2 cruises, kind of similar to this. So I was out for Monterey Bay, the Greater Falerons. I forget what was the other one. It wasn't Channel Islands, I don't think. No. But yeah, it was great. I was out for a shorter time.
Maybe Cordell Bank.
Oh, yes, Cordell Bank. Yes, that's what it was. Yeah, so I applied for the internship and surprisingly got it. The interview was great. And like, I loved everyone during the interview process. And I'm normally really shy, but it was really easy to just talk to everyone during the interview. And yeah, I was really excited to come out. I moved out to LA from Chicago to start grad school. And then in October, I came out for my first research cruises.
What are the sparkly anemone there?
Zoom.
Is that a corallimorph or is that a?
That might be a corallimorph.
Very pretty.
Unless there are anemones with little white spots at their tips, which there may very well may be.
I think it might be. I saw it in the guide here. Let me see.
Yeah, that's a corallimorph. Got that really classic, like, low to the sediment, too.
Okay.
Come wide, Steve. Thanks.
Yeah. Yeah, what
2
computer
black
is that?
corals.
Yeah. That's where we are. We got these gusts that are and it comes down tomorrow. It comes back up again. Eventually. The weight, well, the swell comes up again tomorrow.
Little tiny mushroom coral.
Oh, you got it. Yeah. Yeah, so the swell for next few days, that's why we're getting out here. That's why we're yeah.
You all have another weather event coming in?
Yeah, the swells pick up tomorrow. Right now we're just getting a sustained around 20 knots of wind and gusts to 25, it seems like, which is sort of at the limit, but we're the gusts are we can handle it right now. Plus we're pretty deep, so we know it's coming down, or at least the forecast is for it to come down. So we went in knowing that it was going to peak around this time with some gusts like that. But.
Hey, more rocks.
Yeah.
I know you.
I don't know.
Random rock, or is it?
can zoom there, Steve.
Yeah.
Rock.
Yeah. Different than what's around it, though.
And different little black coral sitting right next to it.
Yeah.
All right. Come wide. Thanks.
If you find yourself sinking all the time, you can back up your Z bias a little if you want it to float more. Go to like 32 or something, it won't sink on you all the time. It's another bad habit to get into, thrusting up. Oh, you're under Argus again.
Yeah, you want to get ahead.
Outside of sleeping, I think a couple of us are going to spend half of tomorrow or half of later today looking up what that weird crustacean was.
Oh, yeah.
Yeah.
Oof.
Hopefully don't have nightmares about it.
Never seen that one before.
Just whenever another stalked crinoid there. I've got a bamboo coral in the corner of the screen.
You are so good at identifying that in the semi-darkness.
To be honest, there's only a handful of species at this depth that would have that striking appearance and not be on rock. So bamboo is a pretty good guess.
What's that floating by?
That's a collapsed larvation house.
Okay. We've seen a couple of those, and I've wanted to ask what not only the collapsed ones, but the full ones. Wanted to ask what they were.
Yeah, they're larvation houses. So they have a little, it's actually not even an invertebrate, it's a protovertebrate, kind of planktonic animal that lives inside, and it spins this big, elaborate mucus house that has multiple lobes that it filters water through. And it has at one section is a little trap. That's what it catches all of the organic matter that it pulls through and eats. And then when it's done, it will swim loose of the house and discard it. And then that floats to the seafloor. And it will just spin up another one there. And they're actually a pretty significant source of carbon transport to the deep ocean. And I think there's also a Bari paper where they turn out to be a significant source of microplastics to the deep ocean.
Interesting.
So is the source of the microplastic, is it just the amount of runoff and the amount of pollution that's in the ocean, and then it's the smallest pieces of plastic particles? Or
yeah, so it's a combination. But yes, a lot of it does come from runoff. So like when we are washing our clothing, that's made of a lot of plastic derivatives. A lot of microfibers wash off, especially when it's a new item of clothing. And then you also have microplastics that are in cosmetic products, such as like face washes and toothpaste. They have like those little micro beads in them. So those are some manufactured, like I think they call them primary microplastics. So like they were created to be that small on purpose. And then there are the microplastics, which are like secondary microplastics that break down from larger pieces of plastic. like if you see big pieces, chunks of plastic floating around, eventually over time, they're going to break down, never ever go away, but they'll break down into small, small pieces. And then even like foam or films from like single-use plastics are considered secondary microplastics too. So yeah, there are a lot of sources where these come from. And that's one thing that people are trying to figure out, like where they're coming from.
If it's so ubiquitous in the ocean, what's the likelihood that we as humans are ingesting it from just eating marine fish and shrimp and you name it?
Yeah, so a lot of labs have looked at different fish. I forget the name of the lab, but this one scientist saw microplastics in every fish that she looked at. Maybe occasionally she'd find some that didn't have any in their tissues. And yeah, so they are being found in the tissues of fish, as well in their digestive, like in their guts. So we are potentially ingesting them, but I don't know what implications this would have on humans yet. That's something that is being looked at. But in terms of some invertebrates and other organisms, it's interrupted their reproduction or ability to feed and development even. But I have seen like some snails that have ingested microplastics off of algae. So that's one reason why I was interested in looking at Macrocystis giant kelp to see if they are vectors of microplastics, because there's this one in-lab study on Fucus vesiculosus algae, which is a smaller algae. It's not kelp, but they did in-lab research seeing if they could retain microplastics on their surface. Oh, look at this. Oh.
Yep. A whole cluster of mushroom corals.
Yeah.
Oh, nice.
Sorry, just noting that down. I can't multitask, talk and type.
You're doing great.
So they
and then
place microplastics in water, so different concentrations of microplastics in water, and then put this algae inside basically and allowed it to soak in the water to see if it would uptake the microplastics, and then they rinsed them off, and basically they just did different trials of this to see if this algae would retain the microplastics on its surface, and they did. And the snail that fed on it ingested the microplastics as well, but they didn't say if it affected the snail negatively, but it did extrude the microplastics out as feces, so that would reintroduce the microplastic to the environment, kind of this continuous cycle. So we'll see. I wanted to see if Macrocystis would carry microplastics.
No. We'll wait the paper that you'll write.
Yeah.
When you
yeah, I'm excited.
Results.
Yeah. It'll be interesting. And of course, there's a lot of other components. So for my kelp research component, I'll be using ADCPs, which are acoustic current profilers, basically, to just take a profile of the current, what's going on in the water, so seeing how the water is moving. So I'll be having an ADCP inside of a kelp forest and one outside to kind of see how the difference of current velocity could be potentially affecting microplastics accumulating or moving around. And I'll also have some sediment traps set out that I'll collect bi-weekly to see how many microplastics are settling along with sediment. And then I'll be collecting blades of kelp to look at those to see if they have microplastics on their surfaces, and then collecting the snails, turban snails that feed on their surfaces, see if they have microplastics in them. I wanted to do sea urchins and some other things, but don't have the time or funding to do all of the things I want to do
Yeah,
for a master's. So
right. And I was going to say there's one person doing this, so pace
yeah,
yourself.
yeah, I have all the ideas, so I just need to focus on a few.
Oh, those are really nice bamboo.
Yeah, what's
Two?
that above
Several.
it? Is that a
Yeah.
Wow.
I think that's a little pronoid coral.
Noad? Is it?
Yeah.
There's another little thing right above that too.
Can we get a bit of a zoom on that,
Steve, you can
possible?
I think
the
there's
one up.
above, yeah.
Yeah.
Or
He'll float up to it.
is that a sponge also to the right? I mean, the left? Yeah, right there on the laser.
It's
Encrusting
something
sponge or a
encrusting on the.
tunicate?
It could be either a sponge or a tunicate.
Yeah.
I think this is a pronoid coral, but it's given the angle, I can't really see any of the polyp structure.
Yeah.
And then there's a, to the right, there's a yellow.
That looks like
That's
a
a
crinoid.
crinoid?
Yeah.
Yeah.
Okay.
Oh, actually, that's not a pronoid. That's a chrysogorgid coral.
Oh,
That's a chrysogorgid.
nice.
Now I'm seeing it.
Yeah, the little fine polyp structure.
Yeah.
They look like little bells.
Yep, like little
From
bells. That's a
this far.
chrysogorgid.
I
Oh,
need to catch
gotta
up.
write this down.
Yep, I'm gonna catch up. Steven, come on. Thanks.
Raj, bridge now.
And look at how dark the
Full
rock
position.
is.
Mm-hmm.
So the layback is about, yeah.
Yep.
About 150 meter layback, so we'll feel that eventually.
Can we get a Niskin taken right here?
As soon as
With
I
the
catch
current,
up, I
I'm not
think.
sure I'll. Yeah. The current, I don't know if I'll get anything out of it, but since we're seeing more coral around in this area.
Yes.
Yes, and this can.
Did you see the fish that just went by?
Mm-hmm.
What was that? Was that again just a grenadier?
I don't know if it's going back. I don't think
Is your rocker set to camera?
it says
I
pretty
think
static.
it
It's
was
already been.
a. Oh, it's not a grenadier. What is it? It's. We
A
were seeing them
brocholid or-
on Davidson.
Yeah, it's all the way back. It's all the way back.
I'm gonna look.
I missed it. Was it a hagfish or something? Or
No, it
Wasn't a
was not a
hagfish.
hag.
a hagfish?
It was my brain wants to say cusk eel, but
Oh,
it wasn't a cusk eel.
A brocholid?
It wasn't a birchulid.
Hmm.
Can tell it's 3 o'clock in the morning. Oh, it was a cusk eel.
Okay.
Okay.
And we can go for Niskin number 3. 1 and 2 have been pulled.
What was that number?
Sample number 68.
think that was Chrysogorgia pinnata. It has that flat leaf-like structure, that flat planar structure, whereas Monticola is the bush.
Gotta catch up a bit here.
Ooh, there's something dark above us. Oh no, it's just the light
Yeah, that's the
or lack of
where-
light.
pointed out into blue water a little bit as we catch up with the ship in Argus.
Yeah.
Yeah, I think our path is not following this bedrock,
All right.
so might have to pull away.
Yep. Left. All right. Roger. See you in my butt camp.
Got another coral there just up and to the right of the lasers that might be another Chrysogorgid or another black. Just looks kind of like a fuzzy cloud since we're back off the cliff.
Yeah, it should be to your left end, but you're we're like way laid back. It's not. Yeah. Bridge nav.
I'm gonna
50 meters north.
drop down and get underneath you.
Yeah, we're trying to get down to this, like the base of this, to follow that up.
All right.
Yeah, I think he just needs to move along to towards 135 as we've been going. Well, then we're gonna have to back down then.
Towards 135. So.
You're gonna have to keep backing away.
All right.
I don't think you'll feel that north move for ages because they're still 150 meters away.
Should I turn around? Should I do this?
The perils of operating at 3,000 meters down.
Roger. I'm coming back. Oh.
That was a very pretty coral, the Chrysogorgia.
Yeah.
I'm hoping as we get our ship adjustment made and move back kind of onto the wall structure, that now that we've seen one, that we get into a few more of them. See if we can get another couple close-up zooms and.
Fly forward. Roger. Yep.
On our next shift, Meredith, the 12 to 4 will be during the ascent, which will take probably the bulk. So I'm just going to have to learn more about midwater pelagic.
Yeah.
Luckily, we have some of those guides as well.
Yep. And it's always you know, you can end up with a long blue water session, but there is almost always something interesting to see during that ascent or descent. I've been lucky enough to see blue sharks, hatchetfish, Humboldt squid.
Oh
So,
my,
yeah.
that would be fun.
There is often things to see. We see a lot of myctophids, which are the little lantern fish come around the vessel.
Roger.
I think you're just still getting dragged 135.
See, and the pelagic zones like that are actually some of the great unknown areas of the deep ocean. We're trying to do more and more work like this with ROVs on the seafloor, but the mesopelagic and the bathypelagic, it's just an environment that, because there's no reference points, it's really hard to work in.
Yeah.
Yeah.
And do you think that work?
Down.
We'll wait for them to do this.
Trying to back down so I can turn around.
Oh, the cliff is long gone. I mean, yeah, I mean, we can't go up and follow that unless like we'd have to backtrack.
Enough.
Still got a lot of 135 to pull.
Coming down. Still one lateral to my right because we're still going 135. Yeah.
We're going to, so I was trying to go down here so we can follow this bit right here. Yeah, we gotta get over there.
Negative. You want to go right. If you have negative wraps, you want to go. Oh, you have positive wraps, so you can go left. You want to.
I think we gotta come up a bit. Market.
Yep. 20 meters off. Yeah.
Starting to get into the down
cam.
cam, yeah. We were in butt cam there for a while. There we go. Yeah, these outcrops, hopefully they kind of keep popping up. It's hard to say how long they go or what the extent is. Seeing a lot of
They
it
seem to
though.
be just, yeah, kind of mixed in along this whole slope face we've been kind of following.
Yeah.
And get into them for a while and then get back out and then get back into them for a while.
It's a very dark
Yeah, another grenadier,
grenadier.
a very dark colored one.
Oh, no worries.
You got 30 minutes to get to that dark red spot on our shift. Can we do it?
Yeah, we can go straight to it if you want.
I thought our
Straight ahead.
original plan is okay with me.
Sorry, what was that?
I said our original plan is okay.
Okay, so we'll just keep coming down here and go up to it. It's hard to say also if that's just, I mean, it's kind of in the direction of a of multibeam. I think the whole feature itself is real, but whether or not that little blip is hard to say. This is over resolved for the quality of multibeam data. That's why it looks all bumpy like that.
Yeah.
Ambitious grid cell sizing.
Could you say again what causes that?
So
Or
we only have a certain amount of pings on the seafloor
mm-hmm.
at this depth. And so if you were to grid at a small enough size, you only have a couple of pings per grid, or maybe even one, or maybe even zero, and then it's interpolated in between. So when there's grid cells with zero data, you just interpolate between the nearest cells to get a value. And so what you can end up getting is a bit of what seems like bumpiness.
That isn't really there.
Yeah.
Yeah.
You've got two different species of black coral on that rock on the left there. There's the heteropathes, and then also what looks to be possibly what used to be called umbellapathes. So they've now changed that genus on me.
Yeah. How about we start going 110 again? How's that sound?
110.
Or 120, something like that. We want to start down here and kind of go up it. That's why I'm. So maybe 135 still. We'll just keep doing that. No. I'll just keep second guessing myself till I call the bridge. Bridge nav. One step, 100 meters, 120.
I think that's just an urchin shell sitting out there on the sediment.
Mm. 7 knots of wind. Spicy.
120, so kind of downslope a little bit.
Yeah, it's kind of,
At least
so
for
we're,
here.
yeah, for here, yeah. And we're also trying to get to see as much of this as we can. So if we start up here, then we only get
Okay.
that bit. So I'm trying to thread the needle between not going too far downslope.
Pulled. I think.
Yeah, it'll probably take a bit for us to gain momentum. So anytime you see anything, feel free to look at it.
Okay.
Yep. Yep.
What are these little white blobs to the right? Oh,
Little,
they're not in view anymore.
we're tracking a sonar target right now. It may just be a rock, but
Oh.
might have some stuff on it.
Straight ahead.
Looks like it. Yeah, there you go.
That's it.
So
Can we have a quick zoom here?
Yep.
On this rock,
Steve.
please? Thank you.
It's encrusting something. Could be bryozoan, could be tunicate, could be sponge.
Yeah.
A little black coral. And I think we have another one of those creepy amphipod things up and to the right.
oh, really? A smaller one?
Just directly right of the black coral, like a little one.
Oh yeah.
Body
Right
shape
there.
looks
Yeah.
similar.
Let's see.
No, we have to look again.
Doesn't have those hooks on its legs that I can see though.
Yeah.
Yeah. Bring down my
Let me know if you
joy
want me
gain a bit.
to come wider.
Oh, I think it's the same thing. I think it's just the smaller one.
And the one we saw before was just massive.
It's got the same antenna back over its head.
Mm-hmm.
Yeah. Get the lasers in the shot. Get a scale of it.
Almost 10 centimeters, not bad with the total leg span.
Roger.
Very similar, if not.
No, I'd say it's the same. Maybe not as bulbous or
Yeah,
but maybe
let's
from this
see.
view.
Same family, certainly.
Yeah, looking at the bathy again, it's possible that this slope we're chasing is artifact. Yeah.
We're moving
120.
120.
Yeah.
So kind of crabbing along again.
Yeah. Yeah. Long contour, roughly.
Better.
Little black coral.
Stock crinoid.
We haven't seen any more of those sea pigs.
No, I haven't seen any in a while, actually. Not since we got into that cliff face.
Yeah. Wonder how much of it, like you said, is the slope versus the depth. We have come up a bit. We're at 3,050 meters.
Actually, in the last little while, we haven't had really any holothurians at all.
Yeah.
Maybe the cliff is acting as some sort of barrier.
Yeah. They roll off.
I say that, and I think we've got one straight ahead, but I think it's one of the ones that swims. Unless that's just a jelly.
Yeah.
Oh, the dark thing that is.
Yeah.
Mm-hmm.
I think it's just a jelly, and I just have a weird camera angle on it on this screen. Tom Leidig has joined us in the science chat.
Howdy, Tom. Oh yeah, he's on soon.
Yep,
Taking over with Chris Caldo.
Oh, we're getting Chris Caldo.
Yes. We have a sighting of Chris Caldo.
To make sure I tune in.
Sailed with Chris several years ago. And Meredith, actually, also